Okay, students, welcome back to the course. Today we will be talking a little bit on fundamental aspects in terms of what are the method used to analyze the DNA. If you're talking about DNA in terms, whatever we are discussing in the unit, we're talking about genomes. At the same time, you're looking into aspects of how do you manipulate the genome, how do you sequence the genome. So if you're talking about DNA, you're talking about one concept called genomic DNA, and one concept is called plasmid DNA, because when you chop the genomic DNA using shotgun sequencing, you have to put them into clones, cloning and back clones, small clones, plasmid clones. So you need to know about plasmid DNA also. How the plasmid DNA looks different from genomic DNA. What are the methods that usually analyze? And then in terms of one more aspect is that we already know that there are single nucleotide polymorphism among human populations. So basically saying that in a given location, one person to the other changes by one nucleotide only. So if taken that one nucleotide change given location, how do you analyze that DNA in terms of the method? How do you quantify that? Rather visually, phenotypically looking at the person, but you need to go into the genotype, isolate the DNA, sequence the DNA if needed, and then check on the mutation in terms of substitution or indel in terms of insertion or deletion. Where is it? Use the sequencing method to analyze it. So one more concept on DNA fingerprinting. It's one of the basic concepts. We will see that what is DNA fingerprinting. Basically saying that the polymorphism between two humans can be fingerprinted, or can be said that a person X from person Y, just like the printing on the unique fingerprints, whatever the patterns on the fingers, they might have a specific DNA patterns also. That we will see why the bands will come up different, why they will have different fingerprinting or DNA printing options. We will, or DNA profiling we call it as. We will look into that. So in the next few slides, 2 slides, I will try to summarize the Unit 3, because given the syllabus, the Unit 3 constitutes close to 15 hours. So we already started the Unit 3 from last week, but given the content, you look into the content here: genome analysis, genome organization and structure. Up to this point, we covered that too little bit fundamental itself only. C values of genomes, repetitive, genetic and physical maps, method of physical mapping. Briefly, we covered it. But when you expand the units again more, you might be encountered with multiple redundant concepts, repetitive or some word change here and there. The concept almost looks same: SSR, ISSR, CAR, AFLP, RFLP. There is only one alphabet changed here. And then here F changed to A, and D, RAPD. So it's bit, it talks much about polymorphisms only per se. The fundamentally of the molecular DNA markers, if you want to understand them better, first you need to understand fundamentally how the polymorphism among humans exist, what is DNA fingerprinting, DNA profiling, how a polymorphism from per se chromosome 1 to chromosome 1 and another person changes in terms of the band patterns, in terms of the microsatellites, mini satellites, or repetitive elements. All those things you need to first understand better. Otherwise, I may take you directly today from RFLP, show some DNA strands moving along here, cut by fragments means I'm saying fragments. It's been digested with some restriction enzymes. So you get 2 bands, 3 bands, 4 bands, done deal. I can say RFLP is done. Cutting with enzymes, look and comparison. Same way I'm saying RAPD here, the A is nothing but amplification. Amplification, random amplification, polymorphic DNA. So I'm saying here cut, I'm saying here I'm amplifying. So basically I'm cutting here without the PCR, just with restriction enzymes. But I am using this specifically technique here, I'm using PCR. Given DNA, I'm amplifying it to analyze the polymorphism in more detail. So basically the polymorphism here differs from here, basically understanding in terms of the cutting it or amplifying it. So if you are amplifying means you are checking on the length, it's shorter or longer, given the DNA sequence from the genome, from the fragments. One fragment may be longer, one fragment may be shorter. So you may use PCR to see the length of it. But in RFLP, the fragment length may be, the genomic DNA or whatever you're analyzing may be constant length. But it may give you short and long fragment given by restriction dilatation or more bands. So the length of the thing, length of the type and variability of the polymorphism given location you are looking, it will allow you to pick one of the method as a molecular marker to analyze the polymorphism in detail. And also this amplified fragment length polymorphism is nothing but similar to this using a PCR again, but there is fundamental difference. We will see into that. Again, micro and mini satellites, as we studied in the last class, where the Google Maps, you go towards that like the same way. The genomic maps, locations will have specific macro satellites and mini satellites. They may be used for again markers, but again they may be again polymorphic between individual. A marker that a microsatellite in a given individual may not be same in the other individual. It may be different, saying that it is polymorphic microsatellite. So you will look into that aspect also. Coming on to short length fragments, simple sequence repeats. The polymorphism, these may be longer fragments, whatever you are looking in these three techniques. Coming on to here, there is small fragments may be there, we will see the length of it. They are called simple sequence repeats. Then what is ISSR? SSR, we need to look into more details. So this is only again half of the unit of half of the portion of unit 3. So somehow the people who designed this syllabus curriculum, I don't know how they did it. That's a common problem as the instructors we are facing. It's okay, it makes some sense here. You can understand that he's talking about the markers for mapping genetic and physical maps, but suddenly he jumps into another approach in terms of in detail analysis and other technological aspects of the same genome mapping. He flips the concepts into different criteria. For example, in situ hybridization, fish molecules, how they can be used to detect the DNA polymorphism without cutting or without amplifying with the PCR. It may be a direct recognition given a DNA location. We might look that, it's a very important concept. And also different markers for amplification. We will see that it may be again based, it's PCR based only. But another concept that's very tricky and important, that's called telomerases. They can be also used as molecular markers. There is a specific tDNA tagging, transposon tagging mechanism there. That also we can look. It's okay, it makes some sense to study this, but again he patches the syllabus into something very diverse and something more into here. Like he mixes viral and bacterial genomes. He wants to talk about general structural features. So whether you want to talk about general structural features of RNA virus or DNA virus, bacterial genome means E. coli, Streptococcus, or whole range, we don't know that. And this we covered a little bit on organization genome. Again, he wants us to look into model genomes in terms of plant, Arabidopsis, rice, and human genome. I think we need to look a little bit more into the chromosome organization, satellites in humans, polymorphism in humans little bit. And then he quickly jumps into X and Y chromosome differences. What is Y difference structure? Why it is unusual from X? He wants to talk about that. And then suddenly out of the bloom, he comes into again chloroplast and mitochondrial genomes. So we need to study this because these are extra chromosomal, right? We will have a genome. If given a scenario, we will have a mitochondria also. Bacteria will have bacterial genome along with extra chromosome elements called plasmids. Let's say given a scenario of mitochondria and chloroplast, he's talking about, he or she who compiled the syllabus, talking about humans or vertebrates where mitochondria is there extra chromosomally. And in plants, given that plants will have chloroplast genome in cytoplasm in addition to the nucleus. And later one more concept of commercializing genomics and polymorphism. So given this diversity of the topics, given the 15 hours, I'm a bit worried and I want to go a little bit slow because the exams are in August. I don't want to rush through this. We will take conceptual wise. We might break and ask for question and sessions after 2 lectures, see where you are standing. We will look into that. But bear with me. Once you understand the general features in today's class, little bit more details on taking one example of RFLP mechanism, a case study, RAPD mechanism, a case study, AFLP, a case study, that might give you more broader understanding of it. Okay, so we will look into that. And one more important worry for me is that the exam pattern may change. You may not have a typical 3-hour exam where much of the choice will be given. you can omit a question, you can take another question. That option may be still there, but I don't know if the university with Ministry of Education, they get an approval for going online exam, then the exam may not be 3 hours. It may be cut short into 2 hours. If it cut short into 2 hours, then there may be a mix of multiple choice questions, mix of handwritten questions may occur, or maybe only handwritten questions may be given. For example, they may ask you to click a link, start your computer, start your webcam, click the link. It will take you another page to download the question paper, just how the way invigilator gives you the question paper. Same way, they may allow you to download the question paper. Then the computer may say, just like the quiz, start time is 0, question paper is downloaded. You have 2 hours to answer everything in white sheets, question by question, just like the answer sheets you write, and then scan and upload that given a portal. At the end of the exam, it may ask you to click a portal and scan the answer paper you have written and submit that to the portal, so that the exam has been administered, the exam has been completed by the student, exam answer sheet has been uploaded. That option, many of the experts are thinking to go in that way. If that option is given only 2 mark to only 2 hour exam, I think the choice may exist or it may not, I don't know that. Or if they mix multiple choice questions and theory together, then if multiple choice questions are there, I think you have to focus on as much as concepts, as much as fundamentals. So otherwise, you might lose a lot of marks in the multiple choice questions. So that's why I don't want to rush through and skip some syllabus. I want to take concept wise a little bit, and then given 15 hours, we might have covering 3 hours now. Still 10 more hours lectures are there, but we will see. The more good we progress, the more quick I can go. So I will expand a little bit more today's class on molecular markers, but basically I will go into in terms of the basic foundation, how you can analyze the DNA for these different amplification fragments or restriction fragments of the given DNA. So we will quickly rush through the basics and see where we stand at the end of the class, and tomorrow we will again meet. So basically the structural genomics we looked in the last class. Given the genome, you can analyze the chromosomes or in terms of the depict some chromosome and put the genes in order on a chromosome based on genetic maps and physical maps. Genetic maps are nothing but you use the fundamental genome like Mendelian genetics, you look for recombination frequencies, and then you assign the genes based on the landmarks here. But on the other hand, if you don't have a genome analyzed well, no landmarks, you can go into physical map based on the sequence assembly and going to find out something and use a software called gene prediction software, ORF softwares. You start to deduce a specific genes on the location. You go in that. In terms of the genetic mapping, we also said in last time that not only using a specific landmarks of the given gene, landmarks of the mutants and wild type crossing overs, you may go into a concept of looking into specific DNA sequences in the given location. The DNA sequences are called DNA markers. They can be used for genetic mapping. The markers are mapped features in a given location of the genome, and they are not part of the gene itself per se, coding region. So that's why the mapped features that are not genes per se, not the coding sequences. Other region non-coding sequences, we have a specific types we look. Excuse me, they might have a pattern. They might have a patterns depending on the polymorphisms. That patterns can be used as a markers for DNA markers for specific individual, and you can compare it with that. The markers may look like, one of them is techniques. How do you look into these markers and analyze the given DNA polymorphism? RFLP, simple sequence, mini and micro satellites, and single nucleotide polymorphisms also you can use as markers. Coming on to the simple sequence length polymorphism, SSLPs, there may be two varieties again. Mini satellites, look into this term, variable number of tandem repeats, VNTRs. Microsatellites, simple tandem repeats, STRs. You might have to write a lot of notes and abbreviations because I might ask this in the quiz. Given the lecture till 13, I might ask abbreviation, match this with that. I might give a question in tomorrow's exam. So look into that. These VNTRs are nothing but they're 25 base pairs length each unit, and the unit may be, you're looking like a block here, one block, 25 base pair. The unit may be 10 blocks together, 25 base pairs, one block, that may be 10 times multiplied in the same location because it's a repetitive element. The same VNTR for X human is 25 base pair each unit, 10 units are there. But in my case, I might have the same 25 base pair length, but I might have only 3 repeats. I don't have 10 repeats in my genome because my non-coding DNA or my junk DNA is different from your junk DNA. So if I look that, same VNTR pattern can change from individual to individual. And then if you look onto these 10 blocks, 3 blocks, if you use a PCR technique, the length will be variable, right? 10 blocks give a longer fragment of the amplicon. That may be 25 into 10, 250 base pairs. But if you take the same my fragment, which is 3 here, 1, 2, 3, imaginary I have written here, I might get only 75 base pairs length. So you can analyze me different from the X person by amplifying specific VNTR in the location of the genome, using specific primers to the VNTR and seeing the product in the PCR, analyzing that, what is the length variability. It is one of the concept in random amplification, RAPD or AFLP. We will see that. Same way, based on the sequence uniqueness, you might design specific molecular biology technique to decode the information. Nothing else but whatever I am talking here, the genome looking in written condition here, the code, you've seen that part of the gene or genome here. But if you look the genome in a living tissue or living condition or in vivo, that the genome doesn't look like this. It looks like a highly supercoiled DNA compiled into a very short fragment or spaghetti form, highly supercoiled intertangled DNA, right? The genome looks highly in terms of the tangles. So if you want to analyze the genome in vitro, how do you isolate the genome? And how do you analyze the genome? It's so tangled. Then how I can analyze it to go into specific sequence? In vitro, you use specific technology of nicking the supercoils of the given genomic DNA. You might break at a single cut using a restriction enzyme. It relaxes the coiling of the DNA. Slowly it separates and it becomes linear. We will see how it looks like. For example, how to isolate and analyze genomic DNA, combining these two questions. Genomic DNA usually called gDNA, and what is plasmid DNA given sense, eDNA, then what are restriction enzymes, polymerase reaction. These are fundamental to you, but looking at an application oriented for in terms of DNA markers. So I will fine tune to in terms of these techniques to the process here. So there is two kinds of DNA, gDNA and pDNA. In terms of given natural native conformation, how the genomic DNA looks, it looks always supercoiled, but the plasmid DNA looks in variable forms. We will see that here. Yeah, okay, the slide moved up here. Okay. The plasmid DNA, given the genomic, it exists in 3 different conformations. One is supercoiled, one is open circular, OC, and a linear form. How you can get these forms based on the natural, because you are producing multiple amounts of the DNA through transformation in a bacteria. It produces not only one copy of the circular plasmid DNA, it goes and produces variable amount because we need to expand the given DNA of insert in the plasmid in terms of the bacteria. So you will get a lot of amount. At the same time, the plasmid DNA will be supercoiled in natural condition. Then you can use a specific condition to uncoil it. That becomes open circular, means only one circle. Supercoiled means having a lot of circles here tangle. Open circle means you somehow you release the tension. You will get only one circular DNA copy, and with the circular DNA, you cut it with a restriction enzyme on both strands, a single cut. It becomes chopped off. It becomes linear here. So it's a linear DNA molecule here. So the linear DNA runs through a gel and comes first based on the size and based on the conformation. We will see that in the agarose gel details. So we have discussed about genomic DNA, plasmid DNA, different conformation, how the DNA exists in a given native conditions. So coming on to an example of looking into a disease condition in terms of functional genomics, wild type DNA samples or RNA samples, given a disease cancer DNA RNA samples. How do you analyze them? First point, before any analysis, you need to extract the nucleic acid from the given sample. So the first step in terms of any genomic analysis or DNA analysis, extract the DNA. Next, you want to go in terms of analyzing the DNA using restriction enzymes, or you're cutting it or you're amplifying it. You're cutting it, you will go with restriction enzyme digestion. You're amplifying it, you go with the PCR, and you have to design primers specific to whatever you're looking for, then amplify, and the amplified DNA later analyze it. Either you cut or amplify, use agarose gels to analyze the DNA. That we will look quickly in this slide, in the coming slides. And given the, in terms of the application of genomics and genomics studies, that is the end of the chapter, we might have to come across and think of in terms of applications of analyzing DNA and in terms of genomics. So basically, you're looking in several tissues here where you get a DNA from different samples. For example, you can get a sample from a specific tissue, liver, heart, or brain, or you can get the DNA from the blood. And each tissue and each sample will have different extraction kits based on the breaking of cell membrane or different cell constituent components. Ultimately, from given a tissue, you have to isolate the DNA in purest form. The form in DNA is measured in terms of the spectroscopy, where 260 over 280, hopefully you know that, you need to get a value of close to 2 or 1.7 to 2 is a good quality of the DNA. The 260 picks the nucleic acid, 280 nanometer picks the protein contamination. So the less the protein, the higher quality of the DNA. So basically you should have a highly good extraction kits. You need to isolate the DNA in purest quality. So based on the cell, based on the tissue, different kits can be used. And just look in the samples here. Samples will only, where the DNA is coming from, will only give an idea about application of genomics in terms of the polymorphism at the end of the unit 3. So tissue got from cancer tissue, blood, specific cells, plant cells in terms of agricultural, and viral and bacterial in terms of infectious diseases, and forensic DNA is mainly in terms of used for discussing in the concept of suspects. You look for DNA profiling, finding a thief or finding a murderer who committed the crime. You use forensic, and then some other studies. In terms of buccal DNA, you have a disease, for example, COVID-19, you need to get the samples of the genomes from that. If a mixed sample of the COVID-19 or the virus along with yours, you need that sample. Strict quarantine procedures will be used here. So if you're cutting the DNA, you use restriction enzymes, that's fine. But if you are amplifying the DNA, again, depending on the length, depending on the type of the DNA, plasmid DNA or genomic DNA, you need to go in terms of this chain reaction. Fundamentally, again, you extract the DNAs based on the concentration, based on the quantity in a given reaction. You just put a specific different samples here and then amplify the samples using a PCR. Fundamentally, it goes for denaturation or linear elongation. At the end, the smallest, whatever smallest quantity or smallest amount available of the start input of the DNA, you amplify it into multiples. But again, you are not amplifying it to some other DNA, you are amplifying exactly the same copy of the DNA in multitude. That means the quantity here you start with 1 nanogram. At the end of the polymerase chain reaction, depending on the cycles, the same DNA will be amplified in terms of the either nanograms, either micrograms or picograms quantity. Because if you are analyzing the DNA for restriction digestion or some other sequencing purposes, you might need a quality and quantity of the DNA. Quality should be good, quantity more to do more restriction digestion, and then send the sequencing. So you analyze basically the DNA usually with agarose gel electrophoresis, nothing but a gel that separates the DNA based on the size. We will quickly go into that. Agarose gel electrophoresis, and then before that, you look in terms of the genomic DNA isolates from the blood. It is coming from the blood, and it's coming from DNA different extraction kits. Given the company, they may use different kits here. But I want to just look in the gel, look in terms of how the genomic DNA looks. Here is all the samples of genomic DNA from the blood isolated by different kits. And here is the control marker M2 landed loaded in this well here, loaded in this well here. And look the distinction of the genomic DNA from the ladder. The ladder is concrete crisps of different sizes, regularly analyzed, arranged by the size of the sample here, size of the marker. Small size goes quickly in the agarose gel, larger size stays back in that. Same way, their distinction, and also the bands look equidistant and equi-concentration in terms, because it's a kind of concept called equimolar mixtures, highly equimolar mixtures. Moles are not different, we will see that. The genomic DNA per se looks quite distinct because it's supercoiled form, and given the conditions, it always looks like that. If this genomic DNA treated with restriction digestion and more cleaning, it looks more crisp and linear. We will see that in the next slides. And when you are talking about genomic DNA, the sizes here in kilobases, you are talking about 350 kilobases. You are talking about 23 kilobases, means 23,100 base pairs is the length of this specific genomic DNA fragment here. So if you isolate genome, you are looking at large DNA fragments. But if you are looking for genome sequencing and shotgun sequencing, you may not analyze the whole. You might have to cut short the DNA. That we will see further. Plasmid DNA we looked in. Again, the plasmid DNA several forms. If you look the plasmid DNA in the agarose gel, it may exist in many forms. One molecule, it will have different forms based on the conformation. Supercoiled in general, that is here. Circular nicked is single, linear cut by the restriction enzymes. Basically, the supercoiled is nothing but a phone, right? You are looking at the coils of the phone here. It's highly coiled. If you stretch one coil with one finger, you stretch some part of it, there will be a high tension will be created in a given other locations. So that tension will allow the genomic DNA go and migrate in this kind of form. That's why you do this spreading of the bands, because you are not looking at a crisp DNA here. You are dragging the DNA through the gel. You are basically pulling it from that string here, super coiled. It looks like that you are coiled me at one point where the old DNA is there, but I am stretching in other position because I have a super coiled form. There will be other stretches, there will be friction, and there will be a kind of force there. That's why you look like that. But you cut it into linear, you will get a single linear band, and you chop it with either circular one single or you chop it with a linear restriction enzymes. That's the way the genomic looks quite different in terms of supercoil. And also, if you look in terms of the gels, the band intensity of the gel changes, right? You are looking at three distinct populations here, one band here, second band here, third band here. The band quantity or the staining is high here. So the staining of the bands is directly proportional to the amount of DNA present. It may be micrograms here, it may be nanograms here, it may be 20 nanograms, it may be 200 nanograms or 1000 nanograms here. Based on the size, based on the intensity, the band. More DNA in a band gives more intense staining because you are staining with ethidium bromide. The more DNA is there, the probe goes and fits more there. This is really important because when you are thinking of homozygous and heterozygous situation of a given allele in SNP genotyping, having a homozygous, having a heterozygous, or having a mutant alleles of the disease or wild type condition, you can recognize the alleles based on the banding pattern of the DNA coming there, because you are amplifying the same fragment given the same primers. You may have an allele distinct in terms of the band staining from the another allele, saying that it may be heterozygous or it may be homozygous for other allele. So that's why I'm trying to emphasize on how do you analyze. Don't need to go into details of alleles or length of the DNA. Just if I give you a gel, I want to ask you which is homozygous, which is heterozygous given the SNP phenotype or given the RFLP, you should be able to analyze. If you from the gel you analyze the RFLP end result well, then you analyze understand the fundamental concept very well. That's why I'm trying to cover. So one of the same DNA is cut with restriction digestion here. One has been chopped into several fragments, and the fragments go by size, go by intensity, because this fragment may be available in the given population more. If you cut with the restriction enzymes, a pool of cutted restriction enzymes, DNA from a restriction enzyme X and Y is available more in a given population or go more in a given polymorphism compared to the other pool of DNA molecule cut with other restriction enzymes. This also tells However, humans differ in terms of polymorphism. If my band looks like this, your band may flip. You may get more DNA here. Location flips with that. So that is really important. So DNA electrophores is nothing but separation of the bands by size. Look into the fundamental here, because the DNA has a negative strand, a negative hydroxyl group coming up. Based on that polarity, given that agarose gel, it moves to the positive electrode here from negative. That's why direction is always vertical. You move down. Given the location, cathode is minus, anode is positive. You put the DNA samples of different sizes, different molecules, different mixtures or different restriction digests. You put here, given the time, given the voltage, slowly the bands go and move according to the size, according to variable polymorphism. Then shorter fragments always go, longer fragments stay in that. So this technique, given the gel, given the length, it allows only 500 bases sequences to analyze it well. For sequencing reaction, you cannot analyze the whole genomes. That is one more restriction for agarose gel. So you look into that. This is supercoiled DNA again. You have seen the stretch. This same supercoiled DNA has been used for restriction digestion. This is a double cut. You get two bands from one here, crisp here, and then you get a different enzyme, a single cut gets linear, another different enzyme, single cut gets linear. Here two different enzymes, two bands will come up. This is important to either understand RFLP or understand SNP genotypic. We will see in that. So these are nothing but DNA ladders. They are equimolar, different sense, 2% gel run by a kind of 100 base pairs ladder. You use it as a marker from starting from 100 base pairs. You go to 3000 base pairs. Same way 1 kb ladder use it to up to 10,000 base pairs. You look into that. So concluding in terms of DNA fingerprinting, so how these polymorphic bands given a different human population can be used to identify criminal. For example, in a crime scene, a DNA sample from anywhere has been got and it's been run through identify different banding patterns that given using the common PCR primers for humans. Then suspect 1, 2, 3 have been analyzed here. Only a perfect match with all polymorphism, complete 100% match with the suspect 2. That's why he has been targeted. Same way, more suspects. Same way you look in the DNA, it looks like different banding patterns. Why there is so much variability? Why there is so much banding pattern, banding sizes, band strains? That's why it comes in terms of the polymorphism only show a different DNA fingerprinting.